Good afternoon, everyone. My name is Himanshi Singh, and welcome to today's session. We'll start in 5 minutes so that more people can join in. Thank you. we can start our today's discussion. So before going into the structures of protein, I would like you guys to have a look on the part of paper or the summary which I am displaying. The title of the paper is The Structure of Proteins, Two Hydrogen Bonded Helical Configurations of Polypeptide Chain. This was the first paper in which the secondary structure of proteins has been reported by Pauling and Branson, right? So what they said is two hydrogen bonded helical structures for a polypeptide chain have been found in which the residues are stereochemically equivalent. The interatomic distance and bond angles have values found in amino acids, peptides, and other simple substances related to proteins, and the conjugated amide system in planar. In one structure with 3.7 residues per turn, each carbonyl and amino group is attached by a hydrogen bond to the complementary group in the third amide group removed from it in the polypeptide chain. And in the other structure with 5.1 residues per turn, each is bonded to the fifth amide group. So what we are discussing here, we are talking about the secondary structure, that is the helix of a polypeptide chain or a protein. So when they are saying about 3.7 residues per turn, they are talking about alpha helical structures. When they are talking about 5.1 residue per turn, they are talking about gamma helical structure. So we're gonna see how they differ, but before that, let's just get us familiarized with the secondary structures of protein in general. Okay, so let me share this small video with you guys. Yeah.
Of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid. So an amino acid has a chiral carbon with a hydrogen attached to it, and it has an amine group attached to it as well. And then it has an R group and a carboxyl group. Now I'm going to draw another amino acid right next to it. The left side, where the nitrogen is located, is the N terminal of the amino acid. The right side, that has the carboxyl group, is the C terminal of the amino acid. Now, these two amino acids can react with each other in a condensation reaction where we're going to lose water, and we're going to connect the two amino acids into one molecule. So once water is removed, we could form a peptide bond. This is also called a dehydration reaction because you're losing water. And so this is going to be the new product of the reaction. So let's see if I can fit it in here. So this oxygen is gone. So now we have a nitrogen. Let me take out H2O. We have one hydrogen left over. We lost two once we lost water. So this is a dipeptide because we have two amino acids combined. And so this amide bond that we see here, that is the peptide bond. We formed a bond between the carbon and the nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know. the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now, the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein. And there's two of them that you need to be familiar with, the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the three-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we could see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding.
Right, so it was a very brief overall view of how many different types of structures are there for protein, as you guys are already aware of it. So one of the type of structure which we're gonna cover in today's class is the secondary structure. So as you know, secondary structures comprises of hydrogen bonds, right, intermolecular hydrogen bonds. So here we are particularly talking about a type of secondary structure called alpha helix. Okay, so alpha helix is a type of secondary structure in which, as the name suggests, it is helical in nature, right? So if a helix is there and it is made up of a polypeptide chain or a peptide chain, then each helix or each turn, just like spring has turns, right? So each turn will have amino acid residues. So when Pauling came up with the structure, when he determined this structure, they calculated that around 3.7 residues are present in one turn of a helix, of a alpha helix. Okay, and we'll see how they have come up with this particular number. Another type of secondary structure is called beta sheet. So this is the paper for that, which I am showing here, is the discovery of the alpha helix and beta sheet, the principal structural feature of proteins. So PNAS papers by Linus Pauling, Robert Corey and Herman Branson in the spring of 1951 proposed that alpha helix and the beta sheet, now known to form the backbones of tens of thousands of proteins. They deduced these fundamental building blocks from properties of small molecules, known both from crystal structures and from Pauling's resonance theory of chemical bonding that predicted planar peptide groups. Earlier attempts by others to build models for protein helices had failed, both by including non-planar peptides and by insisting on helices with an integral number of units per turn. In major respects, the Pauling's-Corey-Branson model was astonishingly correct, including bond lengths that were not surpassed in accuracy for more than 40 years now. However, they did not consider the hand of the helix or the possibility of bent sheets. They also proposed structures and functions that have not been found, including the gamma helix. Okay, so this was a commentary in, I guess, 2003 when it came out, that it's been more than 40 years that whatever Pauling, Corey and Branson came up with, the structures of the secondary structures of protein, they still hold true. There were earlier attempts also for determining the protein structure, but all of them failed just because first they were all looking for integral number of residues per turn, right? As in the previous slide I have said that per turn there are 3.7 residues, but people were not ready, and whenever they were trying to deduce the structure, they were so focused on that there has to be a integral number like 3, either 3 or 4. And also they were not able to predict the planar peptide bonds. So if there is a peptide bond, it is planar in nature, so it can actually if you look in the 3D 3-dimensional, then you can have 3.7 residues. So they are Pauling, Corey and Branson with the protein structure. In 1951, Pauling and Corey said that we have been attacking the problem of the structure of proteins in several ways. One of these ways is the complete and accurate determination of the crystal structure of amino acids, peptides, and other simple substances related to proteins. In order that information about interatomic distances, bond angles, and other configurational parameters might be obtained that would permit the reliable prediction of reasonable configurations of the polypeptide chain. This is the X-ray diffraction pattern of DNA, and this on the right side, this is the X-ray diffraction pattern of beta catenin. So in short, stereochemistry is important in determining which helices are possible, and integral symmetry has no role whatsoever. Now if we talk about the alpha helix and gamma helix, when Pauling, Corey and Branson constructed helices with planar amide groups with the precise bond dimensions they had observed in crystal structures and with linear hydrogen bonds of length 2.72 angstrom, they found there were only two possibilities. These two they call the helix with 3.7 residues per turn and the helix with 5.1 residue per turn, soon to be called as alpha helix and gamma helix. When you put constant to the chemical properties, like this is the bond length, which can exist between two residues, this is the angle which can exist between two residues, then after repetitive how to convey this. Okay, so when you fix the bond length and the bond angle, so when you repeat this pattern multiple times, then you come up with a macro structure. So that macro structure which Pauling and Branson did was this. They came up with two type of helices called alpha helix and gamma helix. So alpha helix has 3.7 residues per turn and gamma helix has 5.1 residues per turn. The idea of the non-integral alpha helix had come to Pauling 3 years before, like when he was visiting professor at Oxford University. He got cold in the damp weather and spent several days in bed. He recalled that he was soon bored with detective novels and I didn't have any molecular model with me in Oxford, but I took a sheet of paper and sketched the atoms with the bonds between them and then folded the paper to bend one bond at the right angle, what I thought it should be relative to the other, and kept doing this, making a helix, until I could form hydrogen bond between one turn of the helix and the next turn of the helix, and it only took a few hours of doing that to discover the alpha helix. This is quite mesmerizing, right? So this is the alpha helix structure as it is depicted. It is helical in nature, and between two turns there are total 3.6 residues. So if you look at what these turns, so these turns are the backbone of the amino acid residues or the protein. So this is the C alpha of first, this is C prime of second, then this is C2, this is C alpha of C2, this is C2 prime, then this is C alpha of 3, this is C alpha of 4, this is C alpha of 5. So you can see how the bonding patterns are emerging. So the C prime of first will bond with the I think something went wrong. I hope you can see my screen and hear my voice. So yeah, so you can see that the first carbon is bonded to the fifth nitrogen, right? Similarly, so there's a bonding pattern of I plus 4, then only you will get the alpha helical structure. And this, if you remember, is the Ramachandran plot, and alpha helix lie in this quadrant of the Ramachandran plot. This is the allowed region of the plot. So the basic properties of alpha helix include, as we are mentioning, the per residue turn will be 3.6 residues, right? Translational per residue, because it's a elongated chain like structure, right? So there has to get translation also. So per residue there is 1.5 angstrom of translation, and translation 5.4 angstrom per turn, and per residue provides 1.5 angstrom of translation, and in one turn there are total 5.4 translation. You can easily calculate it, right? If per turn we have 3.6 residues, one residue provide 1.5 angstrom of translation, then per turn how much translation will it require to complete one turn? Okay, so you can easily calculate that. The hydrogen bonding pattern is CNO. I'm sorry, I wrongly said nitrogen here. It will be oxygen of the fifth carbon of the fifth residue, or say I plus 4, CO of first, and then nitrogen of the fifth residue, that is I plus 4th. So if this is ith, then and this will be I plus 4th N, that is the amine side of the residue. This will be the COO side of the residue. Then phi angle and psi angles are minus 57 degrees and minus 47 degrees. The preference of residues in helix they have, so we have 20 amino acids, right? So not all the amino acids will like to stay in the helix. Why? For example, if there are residues, if we talk about glycine is a very small residue, so wherever it fits, it will prefer to fit in the helix structure wherever the helix needs to get some turns, right? So if we talk about proline, so proline is a rigid amino acid, so wherever the proline is present, the helix will be bit rigid. Okay, so on an average the helix length is not more than 10 residues, and then helices are amphipathic in nature, as we have discussed in the membrane proteins and the proteins which are present in the membrane, specifically the integral membrane protein, right? So how we can say that they are amphipathic in nature? When we look at the helical wheel representation, so this is, just imagine this, that you are looking at a helix or a spring from top, right? And when you're looking it from top, you can clearly project all the residues in one dimension. So if you project it, this is the axis of the helix and these are the residues around that helix. So you can clearly see one side of the helix is hydrophobic and the other side is hydrophilic. Hence this is the reason we can say that helices are amphipathic in nature, because one side is purely hydrophobic and the other side is purely hydrophilic. Okay, then if we look at more properties of the alpha helix, so as you know that peptides have a side, right, the N terminal and the C terminal. So if you are saying that the C of the first amino acid residue bonds with the N of the I plus 4th residue, then at the end of these helices there will be N terminal which will be free and the C terminal which will be free, due to which there is a net dipole movement in the alpha helix. Why? Because the C terminal and the N terminal, they are opposite and they are one on top of each other, giving to the cumulative dipole movement, correct? And it is towards C to N side. Talking more about the helix itself, I'm gonna repeat the particular video which we have shared in the last class, but as we were not much aware of the alpha helix, it was pretty difficult for us to understand. Now as you have little bit of understanding of alpha helix, I really hope that this video and the properties which alpha helix hold will be pretty more clear to you guys. Okay, let me share it. Yeah.
Hello everyone, in this video we'll talk about alpha helix structure, which is a common secondary structure of protein. If we talk about protein structure, then it can be classified into 4 different levels: the primary structure, secondary structure, tertiary, and the quaternary structure. But this video would focus on the secondary structures. Out of the secondary structures, we have alpha helices, beta turns, and beta pleated sheet. But today's focus would be totally on alpha helix, its structure, its function, where we find them, and how we can study the structure of alpha helix and the factors that stabilize it. So this video is all about that. So if you haven't yet subscribed to my channel, hit that subscribe button and stay tuned till the end of this video. First of all, let's try to understand where do we find alpha helix structures. It can be found in many proteins, such as proteins that are receptors, and very common example is G protein coupled receptor. It has 7 transmembrane domain, and each transmembrane domain is alpha helix, spanning 10 to 12 amino acid residues. Many transcription factors such as leucine, which contains leucine zipper motif, that is also an example of alpha helix. So many transcription factor, receptor, ion channels, and many other molecules might have alpha helix conformation. Now let us try to understand how we can study and gain the information about alpha helix. First, we have to start with X-ray crystallography, which begins with crystallization of the protein. Then there is X-ray crystallographic method, where we obtain a diffraction pattern while we pass X-ray sample through this crystal. From this diffraction pattern, we can convert it into frequency domain to get the electron density map. After Fourier transformation, we get this electron density map. Once we get the electron density map, it's like the overall outline of a puzzle, and we have to fit our molecule of interest and start building model of it. And ultimately that model might give rise to the protein structure or structure of any molecule. So this is the overall workflow of X-ray crystallography, by which we can understand the protein structure. If you want to know more about X-ray crystallography, click the link in the I button. So the biggest feature of alpha helix is that helix, and it was found by Pauling and colleagues around 1940s. So alpha helical structure has amino acid in specific orientation. And obviously you can understand the orientation is a helix fashion, where almost 4 amino acids are there per turn. 3.6 to be precise. So alpha helix has 3.6 residues per turn of the helix. All of these residues that are present in the alpha helix are bonded or interacting with each other via hydrogen bonding. And this hydrogen bonding does not take place in a haphazard manner, and there are certain rules that are governing which residues would form hydrogen bonding. Before coming to that rule, we should understand that from a scattered structure of a protein, that means the primary structure, making a geometrical structure like helix is a long process. It requires a lot of planning and organization to make such a geometrically organized structure. So there must be some rule which is governing these interaction, which helps to stabilize these kind of helical structures. Let us understand this rule. There is a rule in hydrogen bonding which is ensuring this helical structure is stabilized. Let's imagine a particular amino acid from the N-terbitus, and we name it I. Then the next one would be I plus 1. After that, there would be I plus 2, I plus 3, and I plus 4. It turns out the hydrogen bond would be formed between I and I plus 4 amino acid. So the carboxyl group of I and the NH group of the I plus 4 amino acid would form hydrogen bond with each other. Irrespective of the position, this kind of rule is valid throughout the helix. Now let us talk about few special cases when helix is forming. Specific residues such as glutamate are good for helix formation. But if glutamate residues are there in repetition, then it might be bad, because glutamate residues at pH 7 has negative charges, and these negative charges could cause strong electrostatic repulsion and thereby destabilizing the helix. The same goes for the basic amino acids, which are lysine or arginine. They are positively charged in pH 7. That is why they can also undergo electrostatic repulsion if they are very close to each other. But if they are dispersed throughout the structure, it might not be a problem. It would be actually favorable in terms of helix formation. Other amino acids such as proline is also termed as helix breaker, because once proline is present in a secondary structure, it would create a tilt in the secondary structure, as you can see here. And the amide hydrogen of proline cannot be used to make hydrogen bonds. And proline side chain interferes sterically with the backbone. And that is why it leads to a tilt of 30 degrees corresponding to the helix axis. So obviously we can understand if we have a proline residue in the secondary structure, it would be ultra destable. It would be destabilizing the helix. So that is why proline is known as helix breaker. Residues in an alpha helix typically adopt specific dihedral angles, and these dihedral angles are really important to define a particular structural characteristics. And the map of dihedral angle is basically the Ramachandran plot. In this Ramachandran plot, these helix falls around minus 60 to minus 45 phi and psi angles. So helix would be corresponding to this particular region. And there are different regions for other particular secondary structures such as beta pleated sheet or right-handed or left-handed helix. Coming to the helix sense, helix could be right-handed or left-handed. Right-handed helix are the most common ones found in biological system. It's very easy to understand right-handed helix. Try to point your thumb towards the helix axis, and your curled finger's direction would tell you the sense of the helix. Now, alpha helix, which is which has a left helicity or a right helicity, would be found in different locations in a Ramachandran plot as depicted here. Also, there are helix propensities. That means the probability of an amino acid to be found in a particular helix. This governs many factors. That means which of these amino acids might be incorporated to form a helix structure. Amino acids like alanine, leucine, methionine are helix makers. That means if these amino acids are present in a protein sequence, they would readily form alpha helical structures. Whereas other amino acids such as glycine and proline are known as helix breakers. They break the helix, but they are very good in terms of beta turn. That is why helix propensity index tells us by a particular score that what is the probability that one particular amino acid would form a helix. Now let me tell you, these alpha helix structures have a lot of peptide bonds, and each peptide bond might have slightly dipole interaction among them. So alpha helix acquires a net positive charge in one side and negative charge in other side due to dipole interaction. And these charges need to be neutralized in order to gain stability. And this is done by specifically negatively charged residues in the N terminus and positively charged residues in the C terminus. Positively charged residues such as arginine and lysine would help to neutralize the net negative charge, whereas negatively charged amino acid would help to neutralize the partial positive charge due to dipole. And this ensures the helix becomes stable. So let's just summarize the factors which are affecting helix stability. First of all, electrostatic repulsion. Second, bulkiness of adjacent R groups of amino acids. Hydrogen bonding between I and I plus 4 amino acid is absolutely crucial. Presence of proline or glycine really destabilize the helix. So these are helix breakers. And lastly, interaction between amino acids and the residues at the end determines the helical stability. So the alpha helical structure can be studied nicely with extracrystallography, and this is the best way of studying protein structure because high resolution protein structures can be obtained from this methodology. But other than that, there are very quick and easier ways to gain information about protein secondary structure, such as circular dichroism spectroscopy. If you need detailed information about any of these topics, you can click on the link in the I button. Now, circular dichroism spectroscopy gives us a specific signature of several secondary structures. In fact, circular dichroism spectroscopy is a best method to study secondary structural changes. So for example, alpha helix, beta plated sheet or random coils would have a characteristic signature in a circular dichroism spectra. Now let's say we have an unknown protein and we would try to plot that unknown protein in this particular graph, and we would see it would match with which particular structure. In this case, you can see in the blue broken line, it's our unknown protein. And it is kind of overlapping with the alpha helical structure. So we can conclude that majority of the secondary structure present in this protein would be alpha helix. Coming to a next example, that if we take another protein and plot its CD spectra, look at it. It kind of match with the beta pleated sheet signature. That means this particular protein might have majority of beta pleated sheet in its structure. So these kind of nitty-gritty details about secondary structures can be obtained with the help of circular dichroism spectroscopy. But other than these two techniques, there is a recent development of cryo-electron microscopy, which can tell us more about protein structure. But what is really different about all of these techniques is the resolution. The highest resolution can be obtained by extracrystallography. Whereas circular dichroism spectroscopy is not, it can give information about protein secondary structure, but it does not tell us about the high resolution structure. Whereas cryo-electron microscopy is kind of high resolution, but not as much as extracrystallography. So with that, we had learned quite a lot in this particular video. We talked about the structure of alpha helix, the nitty-gritty details of it, factors that affect helix stability, and ultimately we learned the techniques by which we can study alpha helix structure. So I hope this video was helpful and informative to you. If you like this video, give it a quick thumbs up. Don't forget to like, share, and subscribe.
So this was pretty specific for alpha helix, as I've discussed the basic properties of an alpha helical secondary structure. More or less you guys now know what is an alpha helix, what are its basic properties and handedness and everything. We're gonna discuss alpha helix maybe in more detail because I felt like that this lecture was not well prepared, so I'll try to do better in the next class. In the meanwhile, feel free to ask any questions. It will help me also to formulate my lectures in a way that is helpful for you as well as for me. So I really hope that we can learn together, and we maybe dig a little bit more on the alpha helical side of the secondary structure, and then we'll go to beta sheets or another type of secondary structure which is predominantly present in the biological system. Okay, so thank you so much for your attention and see you in the next class.